Dear viewers, in this session we would be discussing about variations, mutations, recombination, ploidy, isolation, natural selection and evolution in action. This session has been divided into two parts. In part 1, mainly we would be discussing variations and mutations. No two individuals of a species are precisely alike. It has been observed that the offsprings of the same parents or the offsprings of the same litter and even the twins are not exactly alike. These differences between closely related organisms are described as variations. Variations advocate that no two organisms or parts of the organisms are precisely alike. We would be now discussing various kinds of variations. Variations include changes in all morphological and physiological characteristics like shape, size, color, number and relative position of different parts, physiological behavior of different organ systems, mental ability, etc. This can be further categorized into three contrasting sets. Set 1 has meristic variations and substantive variations. Set 2 includes continuous or fluctuating variations and discontinuous variations or mutations. Lastly, set 3 includes somatic or somatogenic variations and germinal or blastogenic variations. We would be studying each of these sets in detail, starting with meristic and substantive variations. The meristic variations include the variations in the number of parts of an organism, as for example, occurrence of 6 digits on hand or foot instead of normal 5 and presence of 6 arms in starfish while normally it is 5. The substantive variations include variations in form, size, shape or color, shape of the nose, ear and eye, the height of the body or plant, the shape of the leaves, etc. Moving ahead to another set of variations, continuous and discontinuous variations. The continuous variations are minute variations which fluctuate on either side of the average condition and differ only slightly from one another. Since the continuous variations fluctuate on either side of the average, they are also described as minus and plus variations. Darwin regarded them as fluctuations and described them to be significant for the evolution and origin of species. The fluctuating variations occur constantly in nature. The larger is the number of individuals exhibiting a particular variation, the less is the deviation from the mean. The greater is the deviation from the mean, the lesser is the number of individuals exhibiting it. According to Darwin, the progressive accumulation of these fluctuations in one direction leads to the formation of new species. The discontinuous variations are sudden and large. These deviate to a great extent from the normal and do not fluctuate from the average, but appear as totally new. These are also described as saltations or mutations. These are mostly stable and heritable, being transmitted to succeeding generations. The discontinuous variations are rare and without any definite periodicity. A few examples of mutations are hornless calf, tailless kitten, thornless rose and long-tailed Japanese cocks. Hugo de Vries described that these play an important role in the origin and evolution of species. Studying the last set of variations, that is somatic or somatogenic and germinal or blastogenic variations. The somatic variations are caused by the direct influence of the environment upon the body of the organisms. These are local changes in the organization of the organisms and are neither inherited from the parents nor transmitted to the offsprings, but are lost with the parent's death. These are also described as acquired variations. The examples are better developed muscles of athletes, loss of any body part due to some accident, small feet of Chinese ladies, and sunburnt complexion of Europeans in tropics, etc. Lamarck, in his theory of inheritance of acquired characters, laid emphasis on the transmission of these acquired variations from one generation to other, but Weismann proved that these acquired variations are non-heritable. The germinal or blastogenic variations occur in the germ plasm of the organisms. The germ cells or gametes develop from the germ plasm and on fusion form the zygote, which develops into the adult. Therefore, the variations from the germ plasm and the gametes are passed on to the offspring. The differences between blastogenic and somatogenic variations are not distinct, and in certain cases somatogenic variations after repeated generations may produce blastogenic effects. Moving ahead, now we would be learning sources of variations. The sources of somatogenic and blastogenic variations are different. Somatogenic variations are caused exclusively by environment such as heat or cold, presence or absence of particular characteristics, scarcity of food, presence or absence of enemies, and so on. And the blastogenic variations are produced by genetic variability. The various causes of variations are: number 1, environment. The environmental conditions directly influence the organisms and introduce various changes in their organization. The heat or cold, presence or absence of vegetation, scarcity of food, presence or absence of enemies, climatic conditions, etc. influence the individuals to a greater or lesser degree. For examples, puppies from purebred parent may all have practically the same genotype, yet each varies in phenotype from its litter mates. The variations may be considerable. There may be a runt much smaller than the rest, but it also has the same genotype as others of the litter. The runt's phenotype is probably because it could not receive proper food while in the mother's womb or after birth. The litter mates competed so strongly that it could not get plenty of mother's milk. It could not grow and develop to the extent as other litter mates. Yet, if it breeds with one of its litter mates, the runt's phenotype does not appear in any of the resulting offsprings. Tower and Agar conducted several experiments to show the effect of lowering or raising of the temperature on the growth of rats and mice. In majority of cases, mice reared in a warm room at about 21 degrees Celsius were found to differ considerably from those reared in a cold room at 5 degrees Celsius. The shape of larvae of sea urchins and frogs is modified when lithium salts are added to water in which they live. When tadpoles of frog are fed on thyroid of mammals, they pass through all the stages of metamorphosis very rapidly, but remain small in size, whereas tadpoles fed on ordinary diet grow to normal size, but take much longer time to complete metamorphosis. Second source of variation is endocrine glands. The hormonal secretions of the endocrine glands, which influence the development and differentiation of various physical and mental characters, are also responsible for causing various somatogenic and blastogenic variations. The last source and the most important is the cytological basis. The cytological or genetic sources of variations can be summarized in the outline form as follows. Cytological basis of variations broadly includes origin of new mutations and new combinations of old genes or recombination. Both of these factors would be studied in detail. Origin of new mutations include gene mutations, chromosomal mutations or chromosomal aberrations, and changes in the number of chromosomes or ploidy. Chromosomal mutations or chromosomal aberrations include changes in the number of genes and changes in the arrangement of genes. Changes in number of genes include two conditions. Number 1, deletion or deficiency, that is loss of one or more genes, and condition number 2, duplication, that is addition of one or more genes. Changes in the arrangement of genes includes number 1, inversion, that is rotation of a block of genes in a chromosome at 180 degree, and number 2, translocation, exchange of parts between non-homologous chromosomes. Change in the number of chromosomes or ploidy involves changes involving entire sets include number 1, haploidy, having only one set of chromosomes, that is n number of chromosomes, and number 2, polyploidy. Each set of chromosomes is represented more than twice. Triploidy, that is 3n number of chromosomes. Tetraploidy, that is 4n number of chromosomes, and polyploidy, that is 5n number of chromosomes. Changes involving the number of chromosomes in one set of chromosomes includes number 1, monosomic condition, that is loss of one chromosome from one set, 2n minus 1. Number 2, polysomic condition, that is addition of one or more chromosomes to one set, 2n plus 1 or 2n plus 2. And number 3, nullisomic condition, loss of both the chromosomes of a pair, that is 2n minus 2. New combinations of old genes or recombination includes number 1, Mendelian recombination and random assortment of genetic material. Number 2, crossing over or exchange of genes between chromosomes, and number 3, introgressive hybridization. Starting with gene mutations. An individual is the product of interaction of his total genotype, and every gene plays a part in the process of development and differentiation of the organisms. A single gene may affect more than one characteristics. This condition is known as pleiotropism. Or a group of definite number of genes may regulate one characteristic, the condition known as polygenes. Whatever may be the case, genes control the characteristics through the formation of enzymes. All the living processes in the embryos as well as in adults are catalyzed by the enzymes produced in an intricate fashion. The information of synthesis of these enzymes are located in the DNA molecules in the form of sequence of nitrogenous bases. A combination of 3 nitrogenous bases codes for one amino acid and is known as codon or triplet code. Several codons arranged in a definite sequence dictate the formation of particular enzyme and finally the development of particular characteristics. Any change in the arrangement, addition or deletion of specific nitrogenous bases in the DNA changes the entire sequence of bases. The insertion or deletion of a nucleotide into a molecule of DNA would shift the reading of genetic message and would alter the codons beyond the point of insertion or deletion. For example, number 1, if the normal sequence of nitrogenous bases in DNA is TACCA TTAG, it will be read as TAC CAT TAG. Number 2, if a nitrogenous base C is added between CC nitrogenous bases, the sequence becomes TACCC ATTAG. It will be read as TAC CCA TTAG. Number 3, if a nitrogenous base T is deleted from third codon, the sequence becomes TACCA TAG. It will be read as TAC CAT AG. Such mutations in which the entire reading of genetic message is altered beyond the point of change are known as frame shift mutations. These result in the formation of a completely altered polypeptide chain. Gene mutations may also be caused by the substitution of one or more nitrogenous bases in a polynucleotide chain of DNA by some other one. Such mutations are known as substitution mutations, and these change only that codon in which a nitrogenous base has been substituted. Therefore, gene mutation is a change in the chemical pattern of a gene caused by extremes in temperature, cosmic radiation, ultraviolet radiation, radiation from radioactive materials, or some other environmental factors and chemicals, etc. Mutation results in the production of a modified enzyme. The modified, that is, mutant enzyme ultimately express itself by affecting a change in the structure, physiology, or development of the organism. Mutations are generally harmful or lethal. However, some mutations are beneficial too, and mutated organism is able to survive and reproduce. It is presumed that the accumulation of useful mutations through ages has led to creation of new species. Chromosomal mutations or chromosomal aberrations. Chromosomal mutations are the visible changes in the structure of chromosomes, either involving changes in the total number of genes or their rearrangement in a chromosome, or the changes in the number of chromosomes. The former is called chromosomal aberrations. The chromosomal aberrations are caused due to some irregularities during prophase, when homologous chromosome in meiosis break and result in the rejoin and rearrangement. These are of 4 types. Number 1, deletion or deficiency. Number 2, duplication or repeat. Number 3, inversion, and number 4, translocation. Number 1, deletion or deficiency. Deletion is the loss of one or more genes from a chromosome and is caused due to loss of chromosome segment. Depending upon the length of the lost segment, the loss of genes involved may vary from a single gene, that is minimum, to a block containing several genes. The loss of chromosomal segment occurs when a portion of chromosome gets detached due to certain reasons and the broken segment without a centromere is lost. The portion of the chromosome carrying the centromere functions as a genetically deficient chromosome. Deletion can be terminal or intercalary depending upon the loss of terminal or intercalary segment. The loss of terminal segment of chromosome is described as deficiency, whereas the loss in the intermediary position of a chromosome is designated as deletion. Since the deletion involves loss of genetic material, it has some deleterious effect on the organisms and is dependent upon the amount and quality of genetic material lost. A deletion might be small enough not to cause any detectable morphological change in the chromosome and might produce harmful effect. But deletions of large size are usually detrimental to the organisms. Whatever may be the size of deletion, it definitely produces mutant trait. Often the deficiency acts as a recessive mutant. Deletion heterozygotes having small deletions are most probably viable but abnormal, whereas homozygosity for a deletion irrespective of its size is likely to be lethal. Number 2, duplication or repeat. If a particular block of genes is represented more than once in a chromosome, it is known as repeat, and the phenomenon of repeat is called as duplication. The repeated section might have come from another chromosome which suffered a deletion. Duplications are more frequent and less deleterious. This do not lower the viability, but do result in abnormalities of structure or function. Significance of duplication. Duplications play a significant role in evolution. As a result of duplication, additional genes are added to the chromosome's complement. These additional genes might get modified through mutations. Under these circumstances, the lethal mutations might get converted either into tolerable or beneficial ones. In addition, the duplications may overcome the effect of deletion, thus reducing their effect to non-lethal. Number 3, inversion. Sometimes the number of genes in a chromosome is not changed, but the sequence of genes is altered by the rotation of the gene block within a chromosome by 180 degree. If a chromosome having gene alignment A, B, C, D, E, F, G, H breaks at points C and F and the middle segment undergoes inversion, the gene sequence in the inverted chromosome is changed to A, B, C, F, E, D, G, H. Behavior of inverted chromosomes. The chromosomes of a pair having identical inversion undergo normal meiotic pairing and are distributed normally during meiosis, that is homozygous. Even when heterozygous, the inversion has no detectable phenotypic effect, but at the time of gamete formation, the normal pairing is disrupted and abnormal cytological configurations are produced. The paracentric heterozygous inversion with one chiasma in the region of inversion results in the formation of an eccentric chromatid lacking centromere and a chromatid connecting the two chromosomes, thus forming a chromatid bridge between the two chromosomes. Thus, at the end of meiosis, there are two non-crossover chromatids, one eccentric chromatid and one dicentric chromatid. The eccentric and dicentric chromatids are inviable. So, only the non-crossover chromatids form the viable product of meiosis in paracentric inversion heterozygotes. Crossing over in a pericentric heterozygous inversion results in the formation of the chromosomes which have duplications for certain genes and deficiency for others. The gametes receiving chromosomes with deficiency are rarely viable. Therefore, the recombination is lowered down in the heterozygous inversions. Pericentric inversions in normal course lead to changes in the appearance of chromosome. Consequences of inversion. Since the number of genes in an inverted chromosome remains the same, the effects of inversion are not so drastic as are the effects of deletion and duplication. The last and the fourth type, that is translocation. In translocation, a block of genes from one linkage group is transferred to another linkage group. It involves interchange of chromosome segments in non-homologous chromosomes. The phenomenon of translocation can be explained by supposing that the two chromosomes having gene AB and CD exchange segments and produce chromosomes AD and CB. Translocation is thus different from crossing over, which involves interchange between homologous parts of homologous chromosomes. Consequences of translocation. Translocation does not involve loss or addition of genes, but simply a rearrangement of chromosome parts. Individuals carrying such rearrangements should be phenotypically normal unless their expression is affected by the position effect. At the end, we would summarize the entire session so far that we have talked on variations and mutations. Variations are morphological, physiological, cytological and behavioral differences amongst the individuals of the same species and the offsprings of the same parents. Hence, no two individuals are similar. Amongst various kinds of variations, the genetic variations are of evolutionary significance. The meristic variations bring change in the number of certain parts of an organism, whereas the substantive variations affect size, weight, shape and color. The continuous variations are small indistinct variations and the discontinuous variations are large and conspicuous. Generally, the germline mutations are inheritable types occurring in gonad cells, while somatic mutations appear in somatic cells and are acquired mutations. Mutations of chromosomes or genes are alterations that are subsequently replicated. At the molecular level, mutations of genes include point mutations, recombination, deletion, duplication, inversion, translocation and gross chromosomal changes. Mutations are an ultimate source of all new genetic variations, inducing heritable changes in DNA that alter gene expression. Thus, mutations are of great importance for evolution.